Well, welcome for another podcast for the Radiation Research Society, and I'm very pleased to have Alba Gonzalez Junca from UCSF, and you're working with Dr. Barcellos-hoff. And we're going to be talking about EMT, epithelial-mesenchymal transition, and its relationship to radiation, cancer, and maybe you want to introduce yourself and actually tell us a bit about EMT. We can move on from that.
Thank you for having me in this interview. I'm very pleased to be here. So what we are studying here is how this space radiation is inducing a TGF-beta-dependent EMT in non-malignant breast epithelial cells. And the interesting thing is that this EMT, it only occurs when the cells are isolated. So they shift their shape, they lose their polarity and their cell-to-cell interactions when you irradiate them and you add this TGF-beta. And that resembles the initial steps of epithelial carcinogenesis, also known as dysplasia.
Dysplasia, okay. And so let's go through the history of this because, as you know, I work with Dr. Barcellos-hoff for many years. I'm very interested in that topic. I think we've been coining this type of phenotype as non-targeted effect. And as we heard many times on this topic is, one big question is what is the dose dependence of this kind of response and also the LET dependence. And so in your case, you are really tackling both questions since you are looking at high LET radiation from space radiation, and you're also looking I'm not sure if you are doing dose response, but can you walk us through, before you work from Dr. Barcellos-hoff's lab, the kind of main discovery that were done on this topic with respect to radiation and dose response and LET?
So what was initially done many years ago, that was 2007, I believe. I was not even a PhD yet. So what was found is that, as you mentioned, there was not a linear dose response, as you can see, for example, with
DNA damage.
DNA damage. So that led us to conclude that this is actually a non-targeted effect, the EMT, because you observe it as very low doses at 0.1 centigrades, 0.1 grays, sorry. And so then you say that this is actually a non-targeted effect. And so what we are observing in my particular case, since that was already proven and I'm building the story on top of this previous work, we didn't do actually dose responses, so we just used 25 centigrays. But we did compare different kinds of particle radiation. We compared iron, we compared silicon, we did protons, and we did gamma controls. And interestingly, we only observed a complete EMT phenotype, meaning that the cells lose their E-cadherin in the membrane, they lost the beta-catenin, but the beta-catenin efficiently get translocated into the nucleus. That's what we consider complete EMT, and that only happened when the cells were irradiated with heavy ions, with silicon or with iron. It didn't happen in the protons, and it didn't happen in the gamma controls.
With the current technique that you developed, or if without the usage of macrophage?
With macrophage.
So I think we may want to, so just to avoid confusion, the past was done without macrophage. It was just cells that were isolated, they were not confluent, and then TGF-beta was added, and then you added radiation. And when you had those conditions, there was a non-dose dependent, non-LET dependence. You would see EMT after a certain level. So it was more of a threshold systemic response, non-targeted effect.
Yeah.
Describe for us, if you don't mind, the new process and why you've done that, and then we can walk through what you just said.
So what we found is that in tissues, cells are not isolated, and we do not observe this EMT when you look at irradiated tissues. So that let us think that maybe the epithelial interactions were preventing the EMT, stabilizing the epithelial phenotype. So we did a slightly different experiment. We did both in parallel. So we either seed the cells, the MCF10As, and we irradiate them shortly after seeding. That's like between 4 and 6 hours. So just let them sit on the plate and you hit them. Or we wait 48 hours, we seed them at a little bit higher density, and so the cells form cell-to-cell contacts, they divide for 2 days, and when you do the same kind of experiment, irradiating the cells and adding TGF-beta, which whatever type of radiation you use, the EMT is prevented. So there's no EMT occurring when the cells form cell-to-cell contacts. And also with the gamma rays, we did the same experiment called irradiating the cells that were in matrigel, so mimicking what would be the epithelial and extracellular matrix interaction, and that was also preventing the EMT to occur with the same conditions as TGF-beta and irradiation. So what that experiment proves is that the cellular interactions, either with the neighbor cells or with the extracellular matrix, are preventing this EMT, stabilizing this phenotype. So that makes us think, well, then how can these irradiated animals have developed mammary carcinomas, as we have published in the in vivo experiments. So then we came with the idea that maybe macrophages, since we see an enriched signature of macrophages and enriched inflammation markers, we postulated that maybe those macrophages were contributing to this EMT. You know, macrophages in normal tissues, their role is to crawl around the tissues, the tissue resident macrophages, and they are able to separate, to disrupt these cell-to-cell interactions to basically patrol around the tissues. So they have the capacity to break the cell-to-cell contacts that are preventing this EMT. So what we decided to do was to irradiate those epithelial cells that were confluent, where EMT was prevented, and then we put those macrophages on top of the epithelial cells. And then the EMT was allowed. When you have the irradiated with heavy particles, you add TGF-beta and macrophages.
So let me summarize. So you saw for high LET an EMT response that was requiring macrophage recruitment, or I guess co-culture in your case. Yeah. But if you did photons or if you did proton under the same condition, you didn't see it.
Yeah.
So that's fairly new compared to what we had seen in the past, where EMT was always happening with radiation.
Yes.
So can you, why is that? Why EMT happens with any kind of radiation when you do it in non-confluent system, and why once you start going confluent, the macrophage seems to only be able to work on the high LET? What is so special about those high LET?
Well, we don't know we don't know that yet, but we believe it can be a crosstalk with other pathways. So, for example, there's been work published where they show that high LET radiation induces Wnt beta-catenin pathway, and we believe that could cooperate with the TGF-beta. But we haven't done this experiment yet.
It's all right.
But it's a hypothesis.
That's your current hypothesis.
Yeah.
So, okay, so I think the one of the biggest need right now in our field, and I think in the field of biology in general, is a smarter tissue system. So using mice is great, but the problem with mice is, well, they're not humans, right? And using cell culture, right? And cell culture, well, we know a bunch of cells growing in a dish either. So how do you go about this? So one nice thing about DMCF10 is their ability to grow in 3D. And so kind of like getting closer to a biomimetic tissue. And with that idea in mind, I was wondering if some of the work you've done, where you're working with confluent cells, could be repeated with 3D cell culture, where you also have, in a sense, a confluent system, except it's more realistic. And so, but it is a bit of a challenging experiment. You need some very good microscopy. So are you guys moving towards those biomimetic tissues, or?
Yeah, actually that's a really good point, and that's something that we wanted to do, actually, to co-culture this, and also adding fibroblasts, which are also in the normal tissue found. But that was kind of too complicated to run it in that particular experiment, but it's something that we're definitely gonna study in future experiments.
I know Mina Bisso at RBNL, since I'm over there.
Oh, yeah,
they're doing some very pioneering work right now, and you may get inspired by this, because they are now able to grow breast tissue from human. So they have like those little tissue from typically reduction of breast reduction, and apparently they can grow those tissue for 20 days in the dish. And so there, you don't have to do anything. You just use a tissue, the native tissue, and you could do this kind of experiment from the same person. Actually, you could chop it off in a way that. Is it something that could be potentially workable for you guys, or?
Yeah, totally. I mean, especially, working with the MCF10As, it allows you to really control the number of cells that you have and repeat these experiments. So take into consideration that these experiments were run at at Brookhaven National Lab with all this particle irradiation. So those experiments need to be run in parallel with all these controls and everything. But it could be very interesting, for example, to use this system to validate our results in a more clinically relevant or human relevant setting. But yeah, I agree.
Okay, so one last thing that bugs me a little bit, I'm sorry, I'm stuck on this, is that why you don't sit with proton and photon, because, so what were the doses you used for this experiment where you did, can you give us the specific LETs and the dose for each one of them?
So 25 centigrades, all of them. The specific LET, I don't remember. I have the numbers on the...
so you did same dose for all different LETs?
Yeah, and then we did the energies were 1 GeV with protons, and then we had 350 for silicon and 600 for iron.
So I would recommend that you have a stronger positive control for photon, at least at home back at UCSF. That's an easy experiment where you hit them with 1 gray, because you want to make sure it's not a dose effect.
No, the dose was the same, 25 centigrades.
Right,
yeah.
So typically for many phenotypes that are dose dependent, we use different doses for low LET because they're not as potent.
Yeah.
So for instance, for cell death assay, you will go much higher in dose than for high LET. So what I'm saying is if you did 25 centigrade for the photon, you may want to consider maybe using a 1 gray
No,
control.
we did 1 gray and 2 grays, not with the gamma, we did it with X-rays. With the stuff, yeah.
And with the macrophage.
Yes,
and
we
nothing.
do not see the EMT until so you don't see the EMT only on the heavy particles.
And for proton, you only did 1 dose.
Yeah.
25 centigrade. You didn't go higher in dose.
No.
Okay, because it's another low LET radiation. Well, I guess then it's puzzling, I would say.
Yeah, it is.
Right. Interesting.
And the other interesting thing that we see is not only this EMT, it's just cells that are losing their architecture. They are just ugly looking cells. But then you want to see, so to progress further into cancer, you have to lose the growth inhibitory response. TGF-beta is a very potent cytostatic. It was initially described as a tumor suppressor, as you know. So then what we observed that was very interesting, and it only occurred in the silicon and iron irradiated cells, is that in the same condition with high LET irradiated cells together with the macrophages and TGF-beta, we observe an increase in the cells that lost their anti-proliferative control by TGF-beta, meaning they were positive for phospho-SMAD2, which is a readout of the TGF-beta activity, but at the same time they were positive for Ki67, which is a marker of proliferation. So that means that the cells that they were sensitive to TGF-beta, they were receiving TGF-beta, they have an intact TGF-beta pathway, they just didn't stop proliferating.
So to go a bit beyond this, what about going back to the animal model now, and instead of using MCF10A, could you envision a process where you actually repeat the same experiment in vivo? So irradiate a gland and then dismantle the gland and test the EMT activity. And that was one question I had for you, if that's something you're considering doing. The other thing is, I don't know if you mentioned that, but you did do a 3D test on the cells that had undergone EMT, and they had what were their ability to grow in 3D after EMT? Were they disrupted?
Yeah, actually they form disrupted this kind of colonies. Instead of forming these nice little round ducts that the MCF10As form when you put them on top of matrigel, they were completely aberrant. They were bigger and they had all these like kind of protuberances, and that's another sign that they are undergoing this dysplasic phenotype.
And it's a persistent phenotype, which is, it's not something transitory. It's actually
no,
they have
it was
switched.
a week after the radiation. We put them on this 3D.
And do you know if eventually they go back to a normal phenotype, or they just basically are shifted forever?
We never did any longer experiments than that.
Interesting. So it's a very important point, because you would think it could be transitory versus it looks like it's a persistent phenotype.
Yeah, well, but when you go to the in vivo, they do develop more tumors and more aggressive tumors, the ones irradiated with the silicon particles. So that's that. So what we are trying to model here in vitro in a more controlled system is what's happening in the initial events after the irradiation. So we believe that this irradiation, it's recruiting and creating inflammation and recruiting these macrophages or activating these macrophages that were already there, and those macrophages, they secrete. In this case, we found that interferon gamma is necessary. I'm sure they secrete a bunch of other cytokines and things, but when we block interferon gamma from these co-cultures, we prevent this EMT and this TGF-beta resistance, showing that in fact this persistent inflammation, it can be responsible to this initiation of this carcinogenesis.
Right. No, it makes sense. It's very interesting. So just to finish on this in vivo idea, it would be very interesting to take an animal and irradiate the animal, because in the mammary gland, because you will get everything that you need. The macrophage will be recruited, you know that, and then you get the tissue out of it. And since it's a persistent phenotype, you got to be able to, even if you dismantle the what's the term, disaggregate the mammary gland, you should be able to see the epithelial cells having gone EMT. So is it a rare phenotype, or do you think it's in the dish? It looks like the majority of the cells are undergoing EMT, right? I'm having a hard time thinking that in vivo you have the entire mammary gland turning into a dysplasia, right?
No.
So, but do you think it would be an interesting experiment to do? Is it something you guys could?
Yeah, it could be interesting.
Because that looks like the ultimate test, right?
To look at why some cells, because you're right. I mean, in the mammary gland, it's not the 100% of the cells, only some of them, and all of them are p53 deficient in that model, in the in vivo model. So some of them, they might get another hit, or it's maybe because the macrophages go closer. We don't know.
So why is it p53? If you take a wild type animal, you don't have this issue, right?
I mean, well, yeah, we can do it. Just that the
like
p53, it's priming the carcinogenesis. So that's the model that we use for the epithelial carcinogenesis.
Right, I think it's a separate thing. I was thinking of a naive animal that is normal. You expose the mammary gland, and then you collect the gland, and you monitor EMT. I think that was the question.
Yeah.
I would imagine maybe this experiment have been done, but it would be really a nice complementary data set.
Yeah, you're right. It could be interesting to see.
Anyway, I think we're out of time. It's fascinating as usual.
Thank you.
And well, good continuation with your postdoc.
Thank you.
Very nice work.
Thank you.
Do you have any other thoughts, or?
No, that's pretty much it. I think
you.
blocking inflammation is the key to prevent this carcinogenesis.